Mycobacterium, leprosy, tuberculosis, ulcerans. Most of the 190 species are actually harmless saprophytes, non-motile, slightly slender, curved rods, small, living in our natural environment, strictly aerobic, capnophilic, with a high GC content in their genome. From domain bacteria all the way to order Actinomycetes, the family Mycobacteriaceae diverges into Mycobacterium and several others, and the genus Mycobacterium splits into tuberculosis, leprae, bovis, avium, kanzensi, etc. Evolutionary morphology unique to Mycobacterium is our mycalic acid that we find in the cell wall. Nocadia also shares this mycalic acid. It's a different type of mycalic acid, but both of them have mycalic acid in their cell walls. Because of this stain, the primary gram stain. So they are gram positive and must be acid fast stained. Colony morphology is this round mucoid that we see here. And we also identify these species by their growth rates. Some of them are very slow growers and others are fast growers. They're exposed, what they do in light. Some of them give this beautiful orange color pigment when exposed to light, so they are photochromogen. Others actually give a pigment when they're exposed to the dark, which are scotochromogens, and some give no pigment at all in either environment, so they are non-chromogens. Your light test. Evolutionary tree from last universal common ancestor 4.5 billion years ago, we diverge into archaea, domain and bacteria domain. Evidence of bacteria 3.5 billion years ago in cyanobacteria. That splits into about 2.7 billion years ago. Actinomyces become our, and then many years later, many billion years later, 66 million years ago, we find Mycobacterium in our fossils of hominids, possibly hominids which co-evolved with the parasite, and that eventually undergoes a horizontal gene transfer of a plasmid into M. kanzensi 6.4 megabases, which splits into leprae progenitor, which becomes our leprae and lepromatosis. Now, kanzensi also splits into Caneti, which at 4.5 megabases, which becomes our tuberculosis and bovis, etc. Our evolutionary tree. RD, which is the region of difference, is actually a gene that codes for our proteins in Mycobacterium and many other bacterium. So we see this protein is a chaperone protease with ATPase. It's a serine protease. And when we put this into the blast, we discover that 15 Mycobacterium retained that gene, including leprae, tuberculosis, avium, etc. When we put in RD7, which is another region of difference, one species, two strains of it, retain the RD region, and RD8 was retained by one strain of one species. If we look at this tree from common ancestor, we see prior to the RD9 deletion, tuberculosis, and after the deletion, Africanum, and subsequent deletions, including RD8 and 7, etc., speciate the organism all the way down to bovis. And this also shows you the region of difference deletions from 1 all the way to 14. So we see that bovis was speciated with the RD deletions, and that was selected for. That was a natural selection within the bovine environment, where there was a genetic drift, where selection was for the mutated genes that were with the deletions. Thank you to our credits.